Right now we're in a forest in New England. It's a kind of typical New England forest here where you have lots of different types of plants, but a lot of trees. And we're here because we're looking for fungi. We're actually looking for mushrooms, which are the fruit of fungi. What I do is I study microorganisms in the environment. And the reason I do that is because I'm very interested in understanding climate change, actually. It turns out that microorganisms generate an enormous amount of greenhouse gas every year. So my lab and all of my research is really focused on understanding how microorganisms work in the environment as a very complex and diverse community of organisms to regulate or determine how much greenhouse gas is released to the atmosphere from ecosystems every year. So most of what you see above ground is dead. Just underneath that thin layer of dead material is basically a whole world of fungi. Many different species living together, actively decomposing this material. Look at all this fungus! Decomposer fungi, they actually will penetrate dead plant tissue with their cells. They will release enzymes into the plant tissue that break down the plant biopolymers, and they will take up the products of those reactions to use for their own growth, and they will respire a fraction of it to the atmosphere as carbon dioxide. We look for patterns in the distribution of fungi on land and their abundance at different points in times, and we try to figure out why those patterns exist, and that requires a lot of research in the laboratory. So that's where the culture collection comes in. We find species that are out in the environment through high throughput DNA sequencing. We can identify them, and then we culture them out. And we do a lot of biochemical tests on these species to figure out what they are capable of doing. Then what we do is we put them together into synthetic communities. We try to assemble the community as it assembles in the environment and see what the whole community does as a collective, as a consortia. Right now, our models of how carbon moves from the biosphere to the atmosphere as greenhouse gas do not include any of this biology of microorganisms at all. The reason that's a little dangerous is because those models cannot predict the future of carbon dioxide in the atmosphere. We've started to think that because microorganisms are responsible for releasing most of that CO2, that by better understanding the microorganisms and how they do that, we can improve the representation of that biology in our carbon cycle models.